my great honor and privilege to introduce today's speaker, Dr. Ken Buetow, right? Dr. Buetow is currently a professor of life science at Arizona State University. He also served as director of computational science and informatics within ASU, focusing on complex adaptive system initiative. The focus of Dr. Kento's lab is on applying computation technology and the cross-disciplinary knowledge to address major biomedical and environmental challenges. And Dr. Buetow also previously served as the director of Center for Biomedical Informatics and Information Technology within the NIH National Cancer Institute. And within his capacity, because his previous efforts in conservation information system or information system research, so I would like to give a little bit more introduction. In his capacity, he initiated and oversaw NCI's effort to connect the global cancer community through community-developed, standard-based, interoperable informatics capabilities that enable secure exchange and use of biomedical data. And Dr. Buetow also designed and built one of the largest biomedical computational efforts in the world till today. So today's talk is Dr. Buetow will focus on personalized medicine. Probably most of us are familiar with personalized interface, personalized system, personalized recommendation, probably personalized news. However, when it comes to personalized medicine, probably most of us have little experience. So we would really like to hear about what personalized medicine is about and how and why it becomes more technologically more feasible today compared with then. So let's welcome Dr.
Thank you very much. Thank you very much for that generous introduction. And in fact, your introduction hits exactly what I hope to do, is to give this audience a bit of an overview of personalized medicine, what it is, but hopefully convince you all as to why this particular program should be interested in, and I would actually argue is the future of personalized medicine. For us to be able to accomplish what is the core of personalized medicine is going to require us to have deep computational capacity and understanding of a variety of interesting and important problems. So let me get started. So what I'm interested in doing is, in the whole focus of my program, especially over the last 10 years, is trying to ride this exciting developing wave of personalized medicine. So let's start with just the basic description. What is personalized medicine? Well, personalized medicine in its simplest form is about delivering to the right individual the right care at the right time and in the right place. Lots of rights here. And in fact, it's actually in contrast to much of what we do in the biomedical system today, which is really not worried about necessarily targeting anything, but in fact, figuring out what the sort of group mean is of some activity and focusing on the average. So we're not actually, for the current paradigm, interested in this tailoring or specification, but what we're interested in is what can we do that on average works. And what I think you'll see as I walk through the talk, the concept of on average is really very complicated in biomedicine, and in fact may not literally apply to anyone. So we hear a lot about personalized medicine. It's in the newspapers, it's in lay press, it's actually in entertainment shows nowadays. And unequivocally, at one level, personalized medicine is a revolution. It's a revolution as to how we're going about bringing in the individual molecular characteristics of both a person's disease. In the field that I've spent most of my professional career in cancer, it's how do we understand the unique characteristics of that individual's cancer, but it's also how do we understand the unique characteristics of that individual. We're all very complex biologic organisms, and how do we see to it that we do interventions that we take in account of the diverse molecular processes and individual variability that underpins each and every one of us. But I posit that aside from the excitement of the molecular piece of personalized medicine, and again, another reason why computational science has a critical role to be playing, is personalized medicine is also a renaissance. So, at least in US medicine, we have a tradition of personalized care. The original relationship in the delivery of biomedical care was a relationship between the care deliverer and the patient, or the individual attempting to maintain an individual's health. This personal relationship had deep knowledge of this person's preferences, their religious, ethnic, cultural background, what their life decisions would be about, and what would be important motivators for them to be adherent to a health path, or what would be important characteristics necessary for them to be successfully engaged in health, as opposed to just, again, some average characterized component that we tried to deliver one size fits all. So what personalized medicine to me about is how do we provide the customized care, and it doesn't necessarily have to mean individualized, so I focus on personalized versus individualized medicine. Most of us are familiar with the concept of buying clothes at a store. Very few of us can afford to have individualized clothes. That doesn't mean that there aren't folks that go in and get individually tailored work. But in certain sense, this is mass customization. So it's the concept that we have a variety of choices that we could use, that we can use and make informed decisions so that they mix and match in order to be able to provide us the best fit for our unique characteristics. So implicit in this is personalized medicine is a big data problem, okay? And I think you'll hear me return to this as a recurring theme as I walk through this presentation. So, you don't have to just trust me. The next revolution in healthcare, according to Forbes magazine, actually according to many different authoritative sources, there's been a recent McKinsey report and other, is the focus of a trend moving toward big data, tools, and systemization of care, where we mobilize the data that's part of the biomedical system and translate through this mobilization into better patterns of care delivery. Now, I'm more than familiar with it since the word big data is a overburdened term. So let me at least start with, when I use the word big data, what's the definition that I'm using in as I walk through this as to what is big data. Well, big data unequivocally is, sort of goes without saying, big, okay? So it's volumes of data. It's large quantities of data that are, that in the simplest definition are essentially beyond the capacity of a single individual, single computer, or a single laptop to be able to be processed in any sort of meaningful way. But if we only focus on the size of data, and I have to say many of my biomedical colleagues who invoke the word big data are only focused on size, and I guess I'll use that at the edge of inappropriate. Size does matter, but it's certainly not the only factor that drives big data. Big data is equally defined by the variety of resources that one has to assemble. And in biomedicine, you'll see as I walk through this, that, if anything, is the big data challenge, is the fact that the data is cast literally to the four winds, and bringing it together in a usable form is a real challenge. But also, to borrow the Gartner definition of big data, it also is about velocity, how we can do things with data in rapid time, and how we can exchange, integrate, interpret, analyze, evaluate it. So big data, as I'm using it, is the elemental whole that assembles these three V's, that we need to be worried about large volumes of data, its diversity, and how we can rapidly manipulate it into forms that add value, that convert data to information and information to knowledge. So let's start with where most of biomedicine has been focusing on big data. Part of the drive to worrying about big data in biomedicine is driven by the recognition that as we move toward the capacity of fundamental, whole-scale, omic level characterization, we are generating big tons of data. So off of an instrument, for instance, that when one does next generation sequencing, the first piece of evaluation necessary to do a whole genome sequence, to be able to characterize all the A, C, T's, and G's of any human biologic sample, it's measured in terabytes. So the raw files that come off the machines are measured in terabytes. The first round of processing reduces that terabytes down to hundreds of gigabytes. And you can see that even as we start to move forward, the condensation or the collapsing of the data ultimately still on any given individual, even if we extract or limit ourselves to only caring about the unique differences between your sequence data and my sequence data, we're still measuring hundreds of megabytes worth of data for any given individual. Now, what you have to recognize is that this is not necessarily a static representation. So we can talk about, 100 gigabytes that we might need, perhaps sampled when somebody is born, but then our genome changes over time. As we age, it varies. Our tissues actually don't all have exactly the same somatic genome. So we may find ourselves having to generate this collection, this volume of data, numerous times over the course of anyone's life. And from numerous samples in order to deal with the heterogeneity that comes from the trillions of cells that actually constitute my individual biologic characterization. The other piece you can see in this, though, is almost definitionally from the very beginning, there's the velocity piece of this. Using current art technology, these aren't the bleeding edge technologies, but you can see just to do these transformations, these ways of summarizing data are computationally intensive. So this is for a single sample. It takes 24 cores and 72 hours to move from the raw data that comes off the originally processed data to having an assembly. So that's for a single individual, one time only. And I'll point out that there is actually no universal consensus yet about how these algorithms work or what is the best algorithm. We don't really have ground truth in this space yet, especially as we move into the variant calling place. A project that I was aggressively involved with, the Cancer Genome Atlas, every one of the major sequencing groups has a different set of tools, algorithms, and calls a different set of variants. So it's one of the reasons right now we can't collapse the data down to this 100 megabytes, is because we still haven't agreed as a biologic community as to what's the correct, or even if there is a correct algorithmic approach. So we have to stay closer to the primary data so that the algorithms can be available to re-extract or re-examine the data necessary to produce the functional annotation that is the molecular characterization in the biologic space. So unequivocally, I think this, by almost any standard, passes as both volume and the velocity piece of the big data problem. And I guess I want to point out that this is not just some philosophic pie-in-the-sky universe, that the notion of doing this and doing this type of characterization is very real. That, in fact, as we are beginning to move forward, what we see is the future is already here. It's just not evenly distributed. And then, in fact, there's any number of groups that are starting at some routine level to perform this. So, for instance, my colleagues in Mayo Clinic Scottsdale are now routinely taking refractory patients who have failed in their first rounds of oncogenic treatment for their cancer, and doing large-scale genome sequencing to try to evaluate, are there other ways that they could potentially intervene that would facilitate having a more successful outcome. What I want to spend a minute, though, is talking about, again, these are off the shelf. This is an ad from the Washington Post that talks about our colleagues at Vanderbilt and their approach to now routinely using molecular characterization as a way to guide the treatment and to evaluate the interventions that should be formed on any individual patient. A definitive and very popular resource that drives this is the mycancergenome.org. I point you all, this is one of those exercises that you actually can do at home. This is an open resource coordinated by Maya Levy at Vanderbilt University. And in My Cancer Genome, what one can do is thank you. What one can do is select your disease. After doing these sort of molecular characterizations, you can choose your disease. In this instance, it's driven around cancer. Choose, in this instance, melanoma. From melanoma, you can see what genes have been characterized for what are called in this literature actionable mutations, or genes that actually have variants in them that are felt to be targets of clinical intervention. So here's the list from My Cancer Genome. You can see the list of ones that have been described. From there, one can then get deep information about what we know about that variant and how one might intervene. You can see down at the bottom here the potential list, I'll bring this up, of types of interventions one could perform and types of drugs that are targeted at that specific molecular difference in this individual, and in some sense what's known about them and how they might drive response. And shown here is literally drilling down on any one of them. You can then see the specific clinical trials, the amount of evidence that's been generated to date to help support, or what is the outcome associated with using this personalized approach to this individual's molecularly defined disease. So this is pretty provocative and happening today at Vanderbilt, at actually many different institutions across, as I mentioned, our colleagues at Mayo, I believe probably just up the street here a little bit at Hopkins, and
one of our the Vanderbilt lead, Mia Levy, was someone who was actively involved with us at NCI too.
Oh, absolutely, and in fact, part of this is driven out of the original semantic work that we built at NCI. So so this is actually real today, and I think exciting, provocative, but I guess perchance that you leave this room thinking, problem's all solved. It's just a little accounting problem. Let me try to actually put this work into the context of cancer biology. And I'll apologize in advance that I'm purposely going to go through a series of slides that are complex, but I think we need to be very careful, both as information scientists, but actually anybody in biomedicine, of the oversimplification of our problems. Okay, so what do I mean by that? Well, cancer is, aside from you might see here where you just sequence a tumor, find a variant, it's pretty complicated. And actually, I will argue a little more complicated than just picking out a mutation. So what do I mean by that? Well, to do that, you have to be a little bit of a, sort of an armchair cancer biologist. So let me give you a quick 5-second or 30-second interview or introduction to the complexity of cancer biology. So cancer, as the watchword here, is a complex adaptive system. All right, what do I mean by that? Well, first, cancer occurs by the development and accumulation not of one mutation, but in fact, over time, dozens to literally hundreds of mutations. So any time we sequence one of these cancer tumors that are part of these molecular characterization, we encounter anywhere from dozens to hundreds of novel variants, mutations that have occurred over the development of the tumor. Moreover, in the accumulation of these mutations, mirroring the very process of evolution, the cancer itself generates a whole new complex multi-layer organ, okay? Shown here by the different types of cells. So it actually creates a complex, three-dimensional new organ within an organ, to be a little glib in the definition of organ, but it changes in dramatic ways the three-dimensional structure and creates a whole new environment for itself that has neighboring cells and neighboring constitution that may or may not have the same genetic constitution as the core tumor itself. So you can see here there's differences in the collection of variants, as well as differences between all of these different stages of development. So you see that you get dozens to hundreds and a complex three-dimensional structure. So when we now go back to our more simple My Cancer genome, which one of these do we care about? Do we care about this one, this one, this one? It's not clear yet what is the key place that we identify. How do we intervene? Okay? Now, just perchance that this complexity wasn't enough for you, because I can see everyone here is going, this is manageable. A couple of, we're IT people, for God's sake. We can handle dozens to hundreds of things. This is not a problem, right? Okay, but these things don't all just occur by a simple process. They occur by a variety of different ways. So one is mutation. So one is you get direct changes to your nucleic acids, your DNA, in the cells, and they manifest that way. But cancer, to be a little anthropomorphic, is cleverer than that. It actually can use the whole range of biology in order to manifest its phenotype. So it can do this by mutation. It can do it by changing the number of copies. So you may not have to actually alter the nucleic acid sequence at all. You may just need to make multiple copies or fewer copies of this particular gene or variant in order to produce cancer. You may do it by changing how it's expressed. So not at the target itself, but something off target actually changes the rate at which you make this particular product. So this specific effect may actually be only indirectly related to the outgoing phenotype. It may be something upstream with cis or trans, even in this instance, that drives what we see. It can be not nucleic acid driven at all, but biology has a complex regulatory structure that is extra DNA, epigenetic. The epi is over and above genetic, that changes the pattern and how things are processed and determines whether things are expressed or not expressed. It can be done by all sorts of things that modify the effects. Right now, one of the current exciting places, especially post-ENCODE world, is that we realize that there are many things that are not protein coding genes that have tremendous effect on the protein coding gene profile, okay? And when one always hears that the genome is 98% junk, well, it is correct that only 2% of the genome codes for proteins. However, most recent studies it's very clear that 80% of that other 98% is doing something, is actively changing from individual to individual. So it's to be discarding it as junk is almost assuredly incorrect. And some of it is being done through other flavors of nucleic acids that we didn't normally consider in the central paradigm or the central dogma. And then lastly, you don't even have to be using proteins at all. What we've learned is that biology is quite clever. There's all sorts of other things besides proteins that can actually drive phenotypes and cause you to get cancer. Okay? So, complicated enough yet? Still manageable, right? Come on. It's just, we just added another layer, and it's added another dimension to the problem. Okay? So, now, one of the exciting things is over the last 2 to 3 decades, we've learned a ton about cancer. And in fact, now it's pressing 10 to 15 years ago, the fundamental biologic processes that are necessary to develop cancer were well described. Hanahan and Weinberg wrote a seminal paper 10, 15 years ago now that indicated that almost all tumors demonstrate these 6 major properties, 5 to 6 major properties: growth factor independence, evading apoptosis, telomere maintenance, cell cycle regulation, angiogenesis. Okay? But while it's really easy to put one of these 2 or 3 word labels up, what we realize is underlying these are complex networks of interacting molecules that drive growth factor independence or evading apoptosis. For instance, evading apoptosis, one of the key molecules we hear is P53. P53 has more than 25 different interacting proteins associated with it. So what we see is that cancer, to alter these processes, can choose not just a single variant or a single target, but in fact, sort of like ordering from a Chinese menu, one from column A, one from column B, one from column C. One can choose in tumor A to pull on this gene out of this network. But in individual B who develops cancer, a different set of genes. An individual C who develops cancer, yet another set of genes. So again, we're manipulating, the cancer is manipulating these complex collections of networks. So you have this underlying complexity that at some level rivals the internet in its interconnectedness and complexity. So those of you interested in true networks, in studies of networks, biologic networks have the same power law characteristics as the other emerging networks that we study. And it's clear that much of the same engineering that makes the internet robust is what makes biology robust. So you can't take out a single node in a biologic system and make somebody sick. Similar to in the conscious construct of the internet, biologic systems through evolution have created similarly robust networks. Okay, now just lastly, perchance that all of this hasn't been complicated enough, right now we've just been focusing on the tumor, okay? But the tumor occurs in the context of a biologic organism. As I mentioned before, that we change over time. We actually just, the DNA I got from my mom versus the DNA I got from my dad varies at over a million different locations. So that's just within me. There's millions of differences at the nucleic acid level from the genetic constitution I got from mom versus the genetic constitution I got from dad. If we survey across this room, we'd actually have a fair world survey, and those numbers actually go up way above a million. So even captured in this room are now millions of differences that we all are largely normal, healthy, at least within the standard deviations of normal phenotype. We all carry those. But which of those are important? Well, we know that these things drive our genetic constitution, drives how we produce blood cells, or how we produce blood vessels, our cellular matrix, our immune response, but it also, that variation, drives how we respond to our environment. Actually, arguably, one of the reasons we have this rich cornucopia variation is so that we as a human species can robustly respond to very, very different environments. Both environment in terms of what you might think geographic environment, but also disease exposures and all of these other things are a rich tool set then that we actually have to be able to maintain. And these again are measured in millions of variations that have now been described. So let's go back to the first comment. Okay, cancer, it's complicated. Maybe it's very complicated. And I guess what the point I want to make in this is we ignore this complexity at our peril. So, as you might all have heard, I mean, there's a great deal of frustration in the biomedical community that even though we've been spending, literally billions of dollars a year in fighting cancer, why haven't we made more progress? Well, it's a really complicated problem. And I will actually argue to this group is one of the reasons that we haven't made more progress is we've saddled ourselves to what's called a reductionist paradigm, where we actually throw away all that variability, we throw away all that complexity, and focus on an overly controlled system where we think we know what's going on in the absence of this much deeper context, okay? So what I would argue is one of the things that we need to do is embrace the complexity of cancer. We need to embrace the complexity of common phenotypes. And cancer, sadly, will be the tip of the iceberg of complex phenotypes that we face today. So even though my focus is in cancer, things like obesity, diabetes, cardiovascular disease, the refractory forms of these diseases we're facing today all have this similar sort of complexity. They don't have simple explanations as to what's driving them. So we need to embrace this complexity, and you can probably see what's being set up here. One of the ways we embrace this complexity is both by consciously not ignoring it in a reductionist fashion, and again, to foreshadow, checkpoint, by embracing the data that's underneath these and figuring out, not throwing away all the other observations that are part of the real world and using them in meaningful ways. So, cancer, a complex adaptive system. In summary, it evolves over time. It has adaptive behaviors as opposed to static. It changes over time. It displays emergent properties. So the bottom line is cancer and almost all complex systems, the whole is more than the sum of the parts. You just can't take apart all the pieces, then reassemble them and get the same. You actually get emergent properties. There's synergy that comes when you reassemble the system. Requires transdisciplinary study. You can't do it by yourself. Despite the fact that molecular biologists might think that they actually hold the keys to the universe, there are other disciplines that are critically important to understanding the complex adaptive system. And again, to transition into what's the true thrust of where I want to be going with this is, it's got interdependencies. All complex adaptive systems have interdependencies in the context of both resources and information, which brings us to what we're talking about today, information. Well, one of the things before I leave talking about, I've spent a lot of time talking about the complexity of the molecular information. There's a ton, though, of additional information over and above the molecular characterization that we have to bring in to create this context. So clearly genome represents big data, but so does the phenome. And in this instance, when I use phenome, I don't think I see physicians in the room. If there are, I apologize, but because they get really upset when you say actually that they're doing phenotyping. But in biomedicine, and if you think in human science, this is your clinical encounter. This is, doctors are describing your phenotype. So think when I use the word phenome here, this is all of that rich cornucopia of clinical information. And what we see in large-scale projects, for instance, like the TCGA, there's been a tremendous focus on making sure we do every possible dimension of molecular characterization, and then maybe collect 10 clinical variables. What we see is that it's not then creating a rich enough clinical context to be able to embrace this renaissance piece of personalized medicine. And then lastly, again, and certainly not least, and I have to say, probably even more daunting than the other two, is to be able to capture what people have been exposed to. Because almost no one actually has monitors that lets you know from birth to death all of the different kinds of things that your body has seen. Although interesting, if you're an immunologist, my immunologist colleagues would argue that we do have an ongoing flight recorder in our system called our immune system, which is constantly monitoring everything that we were recorded to. And maybe one of the ways we'll unlock this, that I won't go into today, is figuring out how to unlock the information locked in the immune system. So the phenome data is spread out everywhere. It's diverse types, clinical observations, clinical laboratory imaging, registry, biospecimens, reference data, distributed sources. It is cast to the four corners. It's in research centers, care delivery settings, but there's no such thing as a care delivery setting. It's in hospitals, practices, laboratories. It's locked in registries. It's locked in industry databases. It's locked in consumer notes, profiles, and other pieces of information. And of course, as I show here, this is just a cartoonish representation of the complexity of this environment. So this data is spread all across the ecosystem. So when we talk about variety in our big data problem, this variety, it's not, unfortunately, it's not that we have this variety all sitting on one database someplace. It's physically and geographically distributed across many, many different places. And of course, over the last 5 to 10 years, as efforts have stepped up to try to facilitate its exchange, arguably we have perhaps made it worse. So as part of the High-Tech Act, the effort that established the Office of the National Coordinator, there was an effort to create regional exchanges. But what they did is now, instead of creating a universal framework where groups could exchange, they've created 50+ new silos. So all those individual silos we had a minute ago have now had another collection of silos generated that facilitate within a local setting perhaps exchange, but have now solidified differences between regions with very little coordination between those individual pieces. So we've now spent $44 billion, that's with a B, hardening the silos that separate the health information, rather than working at how we might actually facilitate bringing it together. Well, what I showed earlier in the slide, the notion of molecular characterization being a big data problem. I think what's emerging, though, that's going to be, that we haven't even begun to anticipate, is next generation imaging is going to make the molecular data look like a happy problem we had that was just a tiny, minuscule issue that we needed to worry about. As I'm interacting increasingly with my colleagues in the clinical setting, there's a tremendous interest in converting our ability to do molecular characterization into non-invasive techniques that are detected by imaging facilities. And those imaging, each one of these image frameworks actually generate as much data as any one of those genomic profiles. But again, if we're going to be doing this in real time, every time you go in for a visit, you might find yourself being molecularly imaged. So we're about to see an explosion of related data, and there's been almost no discussion in the biomedical arena as to how to deal with that. And then lastly, the true, real, big data challenge is people are starting to personally monitor their health. So I mentioned a moment ago that our immune system may be one way that we're going to be monitoring exposures. Well, the next thing that's emerging is real-time personal devices that people put on that monitor their steps, their health, their heart rate, their blood glucose levels, all sorts of other things that have real-time telemetry reporting information into resources that a patient may utilize, but that we actually have no way to bring into the medical system at this point. It just lives off in another island, completely separated. And this estimate here, actually I need to update this slide, I just realized. We're going to well exceed these 2 million devices in 2013. That was a 2012 projection, and in fact, I think there's already over 2 million devices in 2013, and we're only in May. So, okay, so what does this mean into the future? Well, I think this is where computational science hits its sweet spot, where informatics, information technology, we're masters at this. So the concept, and what I find exciting yet frustrating, but exciting in the standpoint that I think we really can make a difference, but frustrating that we haven't done it yet, is in every other sector of the economy. The concept of leveraging the data trapped in the system is not only just not poo-pooed, it actually is the central focus of what's going on in many, many other contexts. So it's transformed finance, it's transformed the business community, it's transformed consumers. But in biomedicine right now, we still trust the instincts of that good family doc and or the panel experts that get together once every, they meet regularly but produce recommendations every 2 or 3 years. And that's what we feel is the best way to leverage the rich resource. I would actually argue that the future is us mobilizing the data that's in these systems and bringing the same technology that underpins Wall Street firms and day traders and hedge fund managers to be making bazillions of dollars by monitoring deviations and trends, to be bringing that to bear in health and in biomedicine. And my personal favorite, where I think the lowest fruit is before we get to even the elaborate models, is to leverage what in fact is doable on a routine level by groups such as Google, okay? So tools that we could bring to bear in the clinical setting by just having the data available are things as simple that could be helping driving care, similar to how Google, when I start to type in a search term, actually knows not only the words, but then uses its indices to find what were the other words that were most commonly used when those words were entered. This is not a rocket science problem. I'm not saying it's not a computational intensive problem, it's a big data problem, but there's actually off-the-shelf tools in the Hadoop suite that are being maintained by Apache that allow you to do much of this work today. The trick is assembling the data, making it usable, dealing with this heterogeneity and variety, and then applying it to show that it has value. Another related evidence so how would this apply in a clinical setting? Well, as I'm a doc starting to enter into my electronic system symptoms, what it would do is actually cue me for other things that I should be aware of. So it's actually a fundamentally different way of doing a prompt system for docs. It's a way of actually saying and prioritizing based on One of the things one learns as a medical student is when one hears hoofbeats, one should think horse, not zebra. So that's what Google is doing here. So what it's doing is when you type in words and start to type in your symptoms, it's giving you what's the most common thing that happens with this, not the most esoteric. And one of the reasons clinical decision systems struggle in medical contexts is they don't know how to prioritize information. Well, what if we did it just simply based on the frequency with which things are encountered and feed that back to people, not in a thing that is obtrusive, but just helps them perhaps in auto-completing. Okay? I think the other place that we could and should be leveraging nearly immediately this capacity is in the context of what Amazon does with respect to product matching. And I'll actually give a concrete example of how we even prototyped doing this type of work previously when we were at the NCI. But as you all are aware, this is a search. I wasn't doing my Falls New shape here. I put in a search, for instance, a book I'm interested in, Too Big to Know, okay? But as you guys are all aware, what Amazon immediately does is, while I'm typing that, finds the other signatures of similar sorts of books and then brings up those signatures, right? So what if we, in this instance, and you'll see I'll give a concrete example, rather than it bringing up books, which again, for commerce, wonderful. I mean, I'm very pleased these types of algorithms exist. But what if we brought up similar patients and brought what the outcomes were of those similar patients, as opposed to just driving what would be good for consumer, good for books, and good for, and use this in a biomedical context. This is all infinitely doable in the context, okay? So I think all of this is actually just driving us toward what I hope will be transformational in biomedicine. As I mentioned before, it exists elsewhere. But this concept of moving us in our scientific paradigm in biomedicine into our fourth paradigm science. And again, this is probably a little bit like bringing Coles to Newcastle to talk about fourth paradigm science to IT-related groups. But just perchance that you're not as familiar with this, the fourth paradigm science view is about starting to enable the concept of data science and making data science a full partner in the scientific endeavor, okay, especially in biomedicine, and recognizing that we actually are now very data rich but information poor. And what it does is, I even modify this whenever you bring up fourth paradigm science. It says complemented normally on this phrase would be hypothesis testing versus data-driven science. I actually refuse to surrender the concept that data scientists are not doing hypothesis testing. I would actually put before you how many astronomers who predicted the movement of planets actually went out and physically moved a planet to see what would actually happen. So, and nobody actually calls physicists or astronomers, oh, they're just doing descriptive science. They're not really doing anything that's testing hypothesis. Well, I would argue we can be just as rigorous in biomedicine, in data science, in constructing falsifiable experiments that are not just descriptive, but it requires us to be disciplined as well. So I refuse to surrender the word that we're not doing hypothesis testing experiments. And again, it's universal everywhere, and it's beginning to be present in biomedicine. So, in this context, fourth paradigm science is data-intensive science, data generated by instruments, simulation, processed by software. Basically, what I like better is it's the world's sexiest job, right? So, at least for the 21st century. So those of you who were worried that you were actually perhaps going to be labeled as geeks or whatever in going into this type of field, well, in fact, you are actually, especially to a group like this, you are actually entering the coolest field, at least if you trust Harvard Business Review, you're entering what is the coolest field that you should be investigating, okay? Well, what I want to do is spend a little time talking about what we're doing at ASU in this space. So part of what we're trying to do is create a new data science instrument to actually enable what I've just been describing and to be able to do this on a routine fashion, to be able to take this diverse data and do something with it. And in order to do this, we, excuse me, believe you need to create a next generation cyber capability. And you'll hear me because I still am a recovering Fed, going back into acronyms. But if you hear me say NGCC, it's just because it's a mouthful to keep saying next generation cyber capability. So what is this? What does this sort of data science instrument look like? Well, first off, for context, we're all familiar with the importance of instruments. All of our major colleagues have tremendous investment in the creation of instrumentation. And in fact, nobody sees this as boondoggle. I would argue that the Mars rover group right now would not actually have stood for having their creation of a rover labeled as infrastructure. Oh yeah, we can't be funding infrastructure. We need to be funding science, okay? So I would actually argue the Mars rover to the astronomy and other communities actually isn't considered infrastructure. It's actually considered science. So I would actually argue we as data scientists also have to step forward and say, you're not funding infrastructure, you're funding science, similar to this. So, and again, I walk into any research lab and see a several $100,000 sequencer. Nobody labels that as infrastructure. That's science to do a next-gen sequencer. So what does our platform look like? Well, it's a combination of physical capacity, logical capabilities, and human resources. And I'll spend a little bit of time walking through these. So it's an elemental whole that includes all of these. Okay, so I want to be crisp because part of the problem we have in ITdom is we tend to separate all these things. Where in fact, I would actually argue that data scientists needs to be considering these as a whole. We need high performance capacity. We need high performance physical capacity that's ultra high bandwidth networks, large scale storage, multiple flavors of computational. We need rich logical infrastructure, software, metadata, semantics, and diverse teams. Again, think of the Mars rover as a metaphor for here. It's got physical capabilities, it's got unique logical capabilities, and all you need to do is see any of those news briefings at JPL, and they have a team of people that sit day and night working on those types of things, okay? So this is our schematic of the instrument that we're creating. It's got all of these physical characteristics. It's got a large-scale data reservoir, a multi-petabyte data reservoir with transactional capabilities, high-performance computing, massively parallel computing components, and big data components. Because each of these, at least until we truly get to the next generation of physical hardware, require separate instantiations. My colleagues at Cisco would say we're there today. I'm not sure we are quite there yet, but so we have physical capabilities, we have logical capabilities, transactional capabilities, analytic capabilities, deep data resources, as well as context. So I emphasize the variety and the complexity of biomedical data. In my humble opinion, unlike the Google problem, where they literally have tens to hundreds of millions of observations, we actually sadly can't derive context from our data itself because we may only have dozens to hundreds of observations. So we need help in deriving context through semantics. So a key piece of our infrastructure is creating that capacity to define semantics and transform semantics around problems. And then rich collections of diverse data. So let me talk about how we're going about creating this. One of them is I mentioned the importance of semantics. I love this T.S. Eliot quote, "Hell is the place where nothing connects." And I fear that's where we stand an awful lot of the time in biomedicine. So we've been working with a group